Check.
Thank you.
Yeah.
Again, so we are recording this meeting. If you have any concern with us, please reach out before we do so at the end of the meeting. As I said, the goal of this meeting is to go over analysis of SLAP2 data. And the agenda is here. So we first want to talk about these experiments, give them a little more context. I want to remind everyone that those are at the current stage pilot experiments, but they are very valuable. And so we'll spend a couple of minutes describing the context of those experiments. And then Carter prepared a number of resources to facilitate analysis. So he's going to introduce those with Python code. And then I think ideally we spend the majority of the meeting discussing how we want to coordinate and what are the key analysis primitives that we want first to look at. And I'm hoping that at the end of the meeting, 2 or 3 subgroup form or people form that would attack some analysis that we think are very valuable. Does that make sense? Okay, so let me introduce quickly those experiments. So those experiments are done in Kasper Podgorski lab. So we are leveraging the technological development done by his group. And they were done with Andrew Maede, that is here, as well as I'm sure I'm forgetting people, but Michael, she also was involved in some ways. And so we collected 3 data sets on the same mouse. So it's single mouse experiments. And we decided to go with the standard oddball stimulus design. The rationale was that this was the simplest of all the 4 session types. And it's also been shown in the literature that learning occurs within 1 session. And in the current state of SLAP2, it's actually advantageous to do single session instead of chronic imaging. It's possible to image multiple times, but it's a little more work, and so we felt it was more judicious to favor single session designs. What else? We iterated the stimulus script as these 3 experiments were collected. So let me bring up the page where those are described. So if you go here, experiment summary, where is it? Oh, I thought I had a page. Maybe it was, yeah, here. Oh, something has been removed from the script. It's okay. So these are this experiment here. So we did 4 experiments. The first was without a mouse. The second was just a quick pilot for the script, so there's nothing worthy to look at here. And so these 3 experiments we're going to discuss today are all done with a glutamate sensor, glucifer 4 inside pyramidal cells in V1. So this is looking at the activity of synapses. And the first experiments was a shorter session, like a 30 minutes long session. And the 2 the 2 subsequent experiments are one a 50 minutes long session. So it's the same stimulus. And the only difference between the second and the third session is we collected animal behavior videos. So we are sort of adding in more and more on SLAP2 to prepare for higher quality recordings. Yeah. What else? So Carter is going to mention there are 4 different stimulus blocking experiments. The first one is orientation tuning. So we do a serious orientation tuning block, followed by the oddball block, followed by orientation tuning again, and finish with receptive field mapping. So it's 4 stimulus block. Okay, maybe is there anything Made or Casper or anyone's that wants to add to this?
The particular experiment that you're looking at now is an ASAP experiment, but that you're
Casper, are you
showing on the
speaking?
screen. Yeah, can you hear me? No?
Somehow
Okay.
I'm muted.
Don't worry about it.
I can hear you, Casper. Is that
Okay.
Okay. Okay. Casper has some technical issues.
No.
But
Jerome,
we are
you
incredibly
might.
thankful for his team contribution here, and so and this
Jerome,
is
actually, I think
a lot of work. So Carter, maybe you're gonna take it from now and explain the notebook that you put together.
Yeah, Casper, did you want to say anything else in the meantime, really quick? Okay.
No, it's fine. It's fine. Doesn't matter.
Yeah. So I want to point out actually. Go ahead, Jerome.
I realized my speaker was on mute, so I was the problem, not you, not you.
Yeah. Yes. Glad we got that squared away. Yeah, I wanted to add that the experiment you're looking at right now, Jerome, is that's actually, we don't have this one on Dandy right now. So we have 3 files uploaded to Dandy, and they're all with the same mouse.
That's right. That's right.
Okay, so I guess I'll just show the notebook here. 1 second.
I'm sorry about my speakers.
No worries.
Must have been embarrassing.
Okay, can you guys see this?
Yes.
Okay, so this is what we have on Dandy here. If you just go to dandyarchive.org and just look up predictive processing, I think this is also linked in the repo.
Yeah.
You should be able to find this Dandy set here. This is where we're going to store all of our basically finalized NWB files going forward. So right now, you see 4 files on here. This one is old, and it's also unlabeled. So the actual 3 sessions we have are these 3. And those are summarized on the tabs Jerome was just looking at. So here is, I have a local version of the website. Let's see. Yeah, so these should be linked also. So if you, I guess not. Just 1 second. There we go. Let's see. Should just take just a few seconds. Okay. So I'll show you where to find the link for these. So if you're looking at this section here, you scroll down, it's linked here. And so it will just take you to that page that I just showed you. And yeah, if you want to look at any other important metadata, this is the page for it. But now I want to go over a little notebook. So we have this notebook here, which looks like Jerome and Alex Meyer made. So you can take a look at this. And then I made this quick and dirty notebook basically just to get people started with analysis. So if you're not familiar with NWB or you're not familiar with SLAP2 data, this notebook will give you a good launching off point. So you should be able to just download this and then run it yourself. And what this is receptive field mapping. So it just basically generates receptive fields from the NWB files on Dandy. And I'm just going to walk through what it shows, just to give it sort of a quick explanation of what is in our files. So this is the code here using PyNWB and the Dandy API just to download the NWB files locally. There's other ways to stream them, but this is probably the easiest. And then you can see our NWB file here. So if you just want to do like a simple browse of the content in the NWB, you can look at this. It's just like a hierarchical data format. The most important sections here are going to be the intervals section, the processing section, and the subject. Subject obviously has the subject information. And like I said, all of the files we have right now are the same mouse. So they'll have the same subject info. The intervals includes the stimulus presentation, which is sort of a table. And then the processing includes basically the segmented images from the session and the fluorescence. Yeah, so to start off looking at the stimulus, sorry, excuse me, as Jerome mentioned, there's basically 4 blocks in the stimulus here. We have some orientation tuning, some oddball blocks, and then receptive field mapping. In these files, those blocks are not really labeled in the table. We will label them in future files. But for this one, you're kind of gonna just have to look at the parameters themselves and figure it out. So here I have code here that actually identifies where receptive field mapping block starts. But yeah, so this is the content in the table. You can see they're just gradings. They have a couple, basic parameters: frequency, X, Y position, orientation. And then this code here summarizes basically those pairs of parameters here, and you can get a good count of that. And this is the position throughout the session. So you can very clearly see that the receptive field mapping block where the position changes starts here. So for the purpose of the notebook, I just cut off the table at this spot and just grabbed the receptive field mapping section.
Very minor point, but
Yeah.
What you're calling orientation, visual physiologists typically call direction, right? I saw that it goes to 2 pi.
Yeah,
Right.
yeah, right.
Yeah.
Yeah, that's just how it's done in our software. But yeah,
Yeah.
good point. I want to point this out here. So you can see that all pairs of orientations positions, or excuse me, all pairs of positions are have the same count in this portion. That is not true for the 2 other files that we have on Danny. There was a small bug leading to one combination of positions actually having no presentations, and then one down here having double. So you'll notice that, and it will, if you're using different analysis code, it might affect your receptive fields. So I just wanted to point that out so people are aware that it will lead these 2 sections to look a little bit off, but not in this file. Okay, so that's the stimulus. Now let's get to the SLAP 2. So that's stored here. You can pull it out, and the image masks of what is actually imaged are stored in the file. So SLAP 2 images 2 planes simultaneously. So those are called DMD1 and DMD2. And these are displayed right here. So you can see basically this portion of the cell that we selected for imaging, these sort of long regions, and then these sort of sub-selected ROIs within them. So I've zoomed in here. You can actually see, here it is. And in this case, I think this is representing just the fluorescence, like the raw fluorescence. Right. So
These
those,
are actually,
that's just,
so what you're plotting here are segmented synapses.
right.
So what was imaged was this plus a few other pixels that were found to be inactive, and then there was a bunch of data reduction, basically data processing that happened that extracted the synapses. And what you're plotting here, Carter, are the masks of synapses that were extracted.
Right. Yeah. So this is not, I want to point that these NWBs files don't have, right now, they don't have raw imaging data in them. It's all processed and segmented. Right. So yeah, that's an important note. Let's see, which we have,
Carter, you have a question from Karim?
Yeah, Carter,
yeah.
a quick question. Are these in the same plane or are these across 3D?
So each of these is a plane, but the planes are not necessarily parallel, right? They're Casper, can you speak on that?
The planes are parallel. They're the X and Y axes of these 2 planes in the coordinates they're presented in. There's an affine transformation between them, and then they're also at different depths. That information's in the SLAP2 metadata. I'm not sure how much of it has made it into the Dandy set. And so if you want that information, this would be a great, we should interact and we can make sure that more and more information makes it into the Dandy sets.
Yes, I need that information, and I also need to know if you can, in your experiment, know where the cell body is.
Oh yeah, absolutely. And it's not hard. I don't know whether we have it for these experiments. For most of our experiments we do, we also have a structural stack of the neuron, which can then be traced, and then we can get things like the cable distance between every synapse and every other synapse.
Okay, cool.
Thank you. Yeah.
Maybe Made wants to speak to the structural stack she acquired.
I mean, yeah, I think for all of these 3 data sets, we did acquire the structural stack. So we should, I mean, as Casper mentioned, we have that information. We just need to trace it.
Yes. Yeah. Yeah. So that information we just don't have packaged in the NWB yet, but we will. And things like the depth and stuff like that, we'll definitely add.
I want to add the top, as I recollect this experiment, the top dendrites here is is a proximal dendrites, and the bottom one is an apical one imaged at the top of the cortical column.
Yeah. Any other questions? Okay. So now that we have a good overview of what the data are, how it's structured, we can take a look at actually what we recorded. So this is basically the entire session, all the segmented ROIs, and it's the DF4, DMD1. So you can see there's a few brief periods basically where there is no measured value. I think that's a product of the processing. But for the most part, you can see it's pretty clear. You can see an obvious trend of the decreasing fluorescence over time, across ROIs. I think that's a product of the segmentation. Yeah.
The ROIs are sorted by some measure of their SNR per DMD.
Right.
So you should expect that the SNR for smaller numbered synapses will be higher.
Right. Yeah. So yeah, you can see that pretty clearly here. And it's really easy to pull this out of the NWB file also. Let's see. I want to note that this is labeled DF over F. But right now, this is actually not the DF over F. This is just the DF. So in our future NWB files, and when we repackage these ones, we will have the DFF and the DF, and they'll be labeled properly.
Nicholas has a question.
Yeah, go ahead.
Nicholas, can you speak?
I think his microphone is muted, so his questions are in the chat.
Yeah, I'll respond in the chat actually to these sorts of questions so that we can sort of do two things at once.
Okay,
Sure,
thank you, Casper.
All right, so now we've gone over the stimulus and the fluorescence. This is basically just pretty quick code to do receptive field analysis. Essentially, it's just taking each position and then within a specified onset delay and window, it's grabbing, I think in this case, it's the mean DF value from that period following the presentation, and then aggregating that to the receptive field in the corresponding position. So you can run it here by specifying the onset delay. Here it's just 0. So we're assuming responses happen very, very quickly. And we're doing it for 500 milliseconds following the presentation. We do that for both DMD1 and DMD2. When you run this, if you download this notebook and you run it locally, it will generate a huge plots directory, which I'll show in just a second. But just as an example, these are just the receptive fields it generates really quickly. So they look pretty good. And you can see they're all pretty much centered in the same spot. Just to give you a more thorough view of what the output is here, I've run it locally on my machine here, and it generates this plots out directory for the output. And you can see here, it's got it for both DMDs. And you can see all the ROFs here, or ROIs, excuse me. Yeah, so this notebook, like I said, is just quick and dirty. If you are looking for more information for your analysis that aren't yet displayed in the NWB, just let us know. Yeah, if you have any other questions or any issues that you discover with the NWBs, feel free to make an issue on GitHub. Any other questions that pertain to the notebook?
Just the physical distance between these synapses, if that is something that is measured from the structural data, would be great.
Yeah, we'll work on getting that.
I mean, you mean because we can give you the scale factor of the pixels in each plane, but you also want in 3D, is what you meant?
Yeah, so if two or more synapses have been imaged across two different planes, and there is some orientation, or oblique, structure of the dendrite, either the coordinates of these synapses in absolute space or relative distance between them, whatever is feasible.
I think again the planes are flat,
Mhm.
as I understood, and so within one plane, if you know the scaling factor in the RI image, you'll know the distance between synapses within that plane. For knowing the vertical distance, we'll need to give you the imaging depth. That would be a good
Okay.
proxy. And
So
then
and we'll do the geometry calculation.
yeah, I think that would be the easiest. And then we'll also the structural stack, we need to somehow figure out a way to share that. We haven't done that.
Do you know how big
Made,
those files are?
I think Made, maybe you want to answer those questions.
I do not remember how large those files are, but I don't think we want to share I mean, it's not necessary to share the raw structural images. I think we just want to get the geometry and the morphology of the neuron and then extract these information like the distances along the dendrites between pairs of synapses and to the soma and like other informations that you can get from the geometry. Yeah, we can start working on that. Get as soon as possible and add that information to this NWB files. But before that, I think, as you mentioned, the fastest thing to do will be just to measure the Euclidean distances in space. So we do know the depth difference between the two imaged planes. And we know the location of the segmented synapses. So that would be like a rough estimate maybe of the distances between the synapses, though it won't be correct because we are not taking into account the cables basically. But yeah, we should, I think with Casper, we should work on including all of these information into the NWB files.
Okay, maybe I had a few words on what I think are potential steps, and then maybe you can wait on. These experiments has 4 blocks, and I think there are a few interesting low-hanging fruits. The first one, I think, was the receptor field. It's an important control, but we wanted to make sure that we were imaging synapses that are looking at the screen. I think there are avenues of analysis there. The difference in tuning between proximal and apical dendrites, I think, is I mean, there have been reports on this, but at this scale, I think we are probably looking at uncharted territories here. And so I think doing some more extensive analysis of receptor fields of synapses across those dendrites in those dataset is, I think, valuable. That would be the first that I see. The second one is we have 2 orientation tuning block, one at the onset and one after the oddball block. And I think looking at the stability of this orientation tuning after an oddball block is interesting. In isolations, the distribution of orientation tuning in synapses is also, I think, an interesting avenue. There have been studies on this, but again, there are so many synapses here that just the scale is interesting. So that would be, I think, some analysis of orientation tuning in independent block and also how stable they are across the session. I think that's worthy of looking at. And then we have the oddball block that has a single baseline drifting gratings and vertical bar with 4 types of oddballs. The alt, the omissions and 2 alternative orientations. And so we have not at all looked at this data. So it's likely that there's multiple things to look at. One is whether the mouse learns it. How probably adaptation is involved, and you're looking at synapses, so you might be able to sort of start uncovering how that works across all of the synapses, whether they are like subdomains or things like that. So I think that's probably, and this is the main analysis goal of the experiment, is to look at this oddball. And so I just foresee that there's this 3 groups: perceptive field, orientation, and then oddball. And probably once we have them together, we want to see how they tell a story together. But those experiments, there's like 30 minutes, 50 minutes, and 50 minutes. So there's 2.5 hours worth of data on 3 different cells, which I think could answer some of the questions really well already. And so that's what I see. Any other people have analysis idea that should be discussed?
One question I have is the order of, relative to what was presented in the paper, right? So we start, if I get that correctly, receptive fields first, the mapping first.
We do orientation
last.
tuning,
Yeah, the
orientation
orientation.
tuning, oddball, orientation tuning, and then receptive field.
Okay, so do you foresee any value of mapping the receptive field first and then doing the rest, just to shuffle any potential non-stationarity in the data as a result of, bleaching or whatever that happens
Well, again,
throughout the experiment?
I would like to discuss the analysis of this data. We could, in the future, decide to change how it's acquired. I agree. But the reason why we put so much effort into pushing it up is that it will be helpful to have a little bit of a data-driven approach. And so analyzing those data set, I think, could inform what you just said, whether we need to make modification. I'm actually surprised that the receptor field came out so nicely at the very end, because it's a 50 minutes long experiment. We had not attempted this before, and so it's actually a good news. I'm curious if Alex has some thoughts, or what I highlighted is good for everyone.
Yeah, so procedurally, right? I'm really curious, right, about whether an oddball, like what happens to an oddball stimulus, whether there's a novelty response that in these synapses that encodes specific stimuli, or whether it's basically how do I put this? Whether it's like evidence of adaptation would be if a synapse that encodes a particular stimulus, right? Um, that hasn't been shown recently, has a larger response, but it sort of interacts with the stimulus, coding of that synapse. And then sort of like a very high level alternative would be that the synapse just also simply codes novelty, and it just goes off when an oddball occurs. Sorry if I'm using imprecise terminology. Like there's very high level, very simple analyses that could be done immediately on these data. And it sounds like, Jerome, you're not interested in doing much of that analysis here. We're hoping that people in the community will contribute, or do people sign up to do particular analyses, or what's procedurally, what are the next sort of steps to go after some of these low-hanging questions?
Well, I was hoping, I mean, one thing we could do, so I'm just also happy to follow any other ideas. But one thing we could do is just list out on the website the few analysis project we just stated, and then the people that are interested could sort of list their names on Moose and coordinate among themselves about executing this analysis. I think that's one avenue. Yeah, so I mean, perhaps the first step is just to sort of identify who is interested in putting work in analyzing this data, and then self-organize this way. I'm just curious, what do you think? Generally on board?
Sounds good. I just wanted to ask, where do you want to, I mean, do you want to do this now in this meeting, or is there some place where we could coordinate this in a written form? Just
Yeah.
Because I mean, I think the categories you laid out make sense, and then people can just like sort by these topics.
Yeah.
I mean, I would be interested in the oddball responses of synapses, for example.
Yes.
And,
well, what I maybe I'll share my screen. So we've done this forum here. So one thing we could do is list out. Let's see. List out a few one analysis project on SLAP2 in this page, and then have the sub bullet point we just described, analysis of oddball orientation, and then if people want to coordinate, you can use the forum to discuss. But I feel like I feel like we should discuss it here. I mean, if someone is interested in analysis, raise your hands, and then so that your interest is known.
Jerome, can you repeat again? You lined out 4 or 5 different separate analysis. Can you maybe repeat those again?
That's right. Analysis of orientation tuning across synapses, because we have 2 orientation blocks. So just looking at orientation tuning and then stability of orientation tuning across the 2 blocks, I think is in itself interesting. Receptor field analysis of apical and proximal dendrites. I think it's open whether they are aligned. And then analysis of the oddball blocks. So I think there are multiple things that could be looked at here, whether the oddball is actually triggering a response. We haven't looked at that. It's likely, and then whether both are different depending on the 4 different oddball types. And then I think that would be a good start. And then once we have done these 3 things, I think we should compare the orientation tuning pre and post with the oddball response, and sort of look deeper into this. But the intent of this analysis would be to influence future experiments. Does that make sense? So it sounds like, Lucas, you're interested. Alex, are you interested in doing some analysis there?
Yes. Yeah.
And I don't know, Karim, is this something that you?
Yeah. So what we're interested in is essentially comparing synaptic responses, similarity or dissimilarity, right? Let's say 2 synapses or more are tuned to the same stimulus, right? and they are separated by a certain distance, either from each other or from the soma. We're very interested in understanding how this distance relationship maps onto or is correlated with similarity or dissimilarity between stimuli, and how far is it gradual, is it abrupt, is it, you know, whatever it is. So that's one thing that we're very interested in doing. That's why the spatial information is very important to us.
Yeah, you could get started there with the Euclidean distance, and then once you have that, we could work with Made to get the stack segmented.
One question for as an outlook. Would it be possible in principle to also do calcium imaging in the dendrites, preferably locally, like proximal to the synapses that we image, or is that a bit out of scope or impossible?
I mean, I'm sure Casper or Made can speak to that.
The red calcium indicators that are available have not lasted very long in these imaging experiments for us. They bleach. It's something that we might be able to fix. We've also, my group has been developing better red calcium indicators. There are some other ones. It's not going to happen immediately. It might happen sort of at low scale in our lab in separate experiments as we sort of figure out how to make it work. But actually, I think all of these experiments, these neurons were expressing a calcium indicator. And basically, the calcium indicator bleaches so quickly that we can only get a somatic signal out of it and only in a subset of neurons. And we just decided to focus on the glutamate signals in these initial experiments for this purpose. Not impossible, probably out of scope for the immediate moment, and then who knows in the future.
Thanks.
Yeah, I think the intent here is because it's likely that some of the oddball responses are implemented at a single cell level, and the field has studied some of the standard oddball as potentially a network mechanisms. And so I think here we would be diving deep in one cells and trying to uncover how much of it could be happening within the realm of the cell. And so I think that would influence the other experiments a great deal. And yeah, I mean, with Alex Meyer and Andrew Bastos, we've shown that some of the standard oddball are really, really ubiquitous across the cortex, and so it's likely that there's something going on inside the cell, in my opinion. And I think we have a good chance with this data to uncover that part.
Especially if we find interesting things in the synaptic inputs and the like patterns and statistics of the synaptic input, that would motivate us to sort of do the extra work to sort of find cells that are expressing enough of the calcium indicator to the dual experiments. But yeah, there's definitely a lot to do just with understanding the input patterns to individual cells.
I mean, according to theories, an important contribution is postsynaptically, so it might be hard to spot in this experiment, because I mean there might be postsynaptic inhibition contributing and other processes. So yeah, but already looking at the synapses is already very interesting. I mean, some of the properties could also be inherited from the inputs, and that would be important to know already.
Totally agree.
I was just wondering, with the stimulus paradigm that we have so far, if we look at oddball response, I mean, we can compare them to the control block, right? But for the temporal frequency deviant, I didn't spot a proper control for that because we do not show any static grading in any other point in the stimulus paradigm. So I was wondering, just from an analysis point of view, what we compare this response to in order to figure out if this response is enhanced or the same, or
Are you thinking about the halt?
Exactly. Yeah.
Yeah, that's a good point. We could, I mean, if this is a good thing to bring up. If I could influence the experiments. We have, I think the orientation tuning blocks are designed to provide a control for the alternative orientations in the alt balls. I agree. The alt is a good point. I don't think we are presenting in control blocks. We should have had it eventually. Also the omission. Yeah. We'll make mistakes. It's fine. We're correct.
At what point, what would the process be for updating the stimulus paradigm? I imagine that we want to sort of cycle through some analysis and then accumulate sort of a list of changes and then sort of flash a bunch of changes for a second round if we do refine the stimuli. I think no doubt there will be refinements to the stimuli, but I think that we want to have chunks of data also that are sort of internally consistent. So what's what do you see as the timeline for that, as the process for that?
Yeah, I mean, in practice, I think we should create issue ticket on the repo, probably, with like these concerns. So I would discuss it in the forum, and then if we agree there's an issue, we should create a ticket, and then we can decide to make a stimulus release, like aggregate multiple issues and then pack it in one. At the moment on SLAB, we are working hard to strengthen the pupil tracking and some of the related things like gamma calibrations and others. So we're fixing that. I think the stimulus design, we could change it. Yeah, probably in the block. I think Casper is wise that there's value in having a more homogeneous set. Yeah. Karim?
Are we imaging in between the stimulus presentations? In other words, is it continuous so that we can at least estimate, for example, if the firings or if the synaptic activity during that time is reflective of the time between the presentation of similar or different stimuli?
Very good question. So the session is semi-continuous. So the last two sessions are 51 minutes long, and we image throughout the entire duration. Due to how SLAP is being triggered, essentially we acquire 30 seconds long movies interspersed with I think a half a seconds of gray, so that we can restart the microscope. So it's 30 seconds, really brief stop. 30 seconds, really brief stop. And it's continuous like that for a whole hour.
I see. So the missing data is just this brief stop.
Yeah, just a brief stop. We actually are going to remove that with Casper and Made, but it was just a technical limitation when we started.
Okay.
Lucas?
Yeah.
Oh, sorry. You have something, Karim, to follow up on that?
No, I was just going to say it is important if one at least wants to get a good idea about how the synapses are responding for the repetition of the same stimulus to build some prediction. It has to at least incorporate some time information within that for how long each stimulus was presented, for how long where the stimuli were separated, and so on and so forth.
You have that info, I think, because the NWB is all registered to a same clock, and so the timestamp that are in the files are relative to the start of the session.
Right. Thank you.
Lucas, you had a related question, or?
Yeah, I want to pick up Casper's point again. So I think one thing we can consider is doing it post-hoc, like after exploring the data and we see there's some things we have to change in the stimulus, but there's also considerations from the hypotheses and analysis that we want to do that we can know in advance, and probably makes sense after forming these groups of analysis to come up with the hypotheses in these groups and the analysis that we want to do, that we plan to do, and if we can do it with the current stimulus paradigm, or if there are changes that would allow to address these questions, then we can do that already in advance and see what stimulus paradigm ticks most boxes so that we can address maybe most of these questions.
Right. I do want to mention we have written an analysis plan that probably cover a number of hypotheses. Some of these might not be applicable to single cell dendritic imaging, but I think many are. For instance, the multiplicative versus additive or other ideas are really applicable here. And so I would encourage to just see pieces of the analysis plans that are relevant in this particular case. Would you agree?
That's a good point. It's just I think in the paper we were at a level of abstraction that was a bit higher, and now that we have the actual stimulus laid out, in code, and we see what's happening precisely. Because I checked in what we proposed, and it's not as detailed as the stimulus that's implemented currently. So I think it's just on a bit different level of abstraction now. We have to go this extra step and make it very precise so that we make sure what we want to test is really in that stimulus paradigm.
Okay. I can help a bit in the project tracking page, I can create the subthreads we just discussed, and then maybe sneak in aspects of the analysis plans that are relevant. And so if you want to make comments on it to strengthen them, I think that's welcome. Use the forum for that. I think it's a great way to discuss it. Does that make sense?
Yeah, that makes sense. Yeah.
Karim, I want to address a question from Sarah earlier.
Oh, sure.
Okay, she said, how does the pupil tracking works in your hands? I assume that the tracking quality depends on the imaging depth, but I'm not sure how you deal with this. Seems to be so pupil tracking is eye tracking. We've acquired one of the last experiment. We actually have eye tracking, a video of the eye. There's some issue of crosstalk that we have to deal with and had an additional filter, but we can see the eye throughout the whole session. And we haven't processed it yet. So I would assume that eventually down the line we'll have the pupil size in the NWB, but this is not there yet. I don't understand the tracking quality depth point. So maybe you want to rephrase your question, Sarah. Okay. Do you use an IR light source or the laser itself? Oh, it's a separate LEDs. Which is why we have crosstalk from the IR at the moment. We just need to add a filter. That's a quick fix. Okay. Karim, you had a question?
Yeah, regarding to something Casper said earlier, which is the next set of stimuli presentations in the passive viewing, the standard mismatch. So right now, as I understand it, in figure 10 of the paper, we said we're going to present the oddballs randomly every 11 seconds or so, right?
Yeah.
Have we thought of a sequence where you present the same stimulus, but then when the oddball happens, you follow a certain pattern of orientation changes? So you could go, for example, clockwise. So you present 4 vertical orientations, for example, and then you follow that with, another 4 of, horizontal orientation, and then another 4 of another rotation of a 45-degree angle. So in other words, we're trying to incorporate some sort of a long time scale prediction beyond the single oddball block, if you know what I mean.
I think that's the sequential oddball that you're describing.
Is it? Which is session 3?
Yes.
No, it doesn't. Well, the way figure 10 is displaying it, maybe I can share my screen here. So this is the sequence mismatch, if you guys see my screen.
Yeah.
So over here, it does not show that we're following a specific sequence.
Oh,
Because
maybe the figure is not clear. I think the intent here is orientation turns, in this example, orientation turns, and then you repeat that cycle over and over again. And then
yeah, so that's different from what I'm talking about. What I'm talking about is really the standard mismatch, which occurs like the top one here.
yeah.
But then at the time of the oddball, you present, you know, a different angle repeated, let's say 4 times or whatever. And then the next block is going to be another rotation clockwise and so on, or anti-clockwise, but you follow a certain pattern of mismatch instead of making it random every repeated block.
So you
Still
want
in
the
the
mouse
standard
to
case.
learn? Is this for learning or?
No, this is just to see if I return back. So let's say after a certain, let's say I sampled 360 degrees with 8 45-degree angles, right? So I present 8 different orientations, and then I return back to the same, let's say, vertical one that you see here in this picture. The question is there any adaptation that happens over that long time scale as opposed to a short time scale that happens within blocks or within a a specific mismatch presentation?
I don't think we've discussed that before. Is that a? I mean, is that a? I mean, you
It
think
does
the
have.
mouse will. These are very long time scales. You expect the mouse to remember.
So how many of these repeated ones exist within a block before the mismatch happens? There are 40 of them, according to what is
There are
stated here,
40
right?
mismatch. There are 40 mismatch, but the number of drifting gratings, yeah, it's semi-random when they're presented, but it's on average every 11 seconds.
So that number of presentations is not fixed for a specific angle.
No. It's
So
random.
sometimes you have 3 of these repeated, sometimes it's 10.
That's right.
Okay.
It's unpredictable.
I think there is some value, at least that over the long term. It doesn't have to be, a change that we need to do right now, but it's something just to explore this idea of, forming the prediction over a multitude of timescales, as opposed to studying the oddball, the mismatch occurrence, which is a narrow window of time, but it could be that there is a longer time scale that's being learned here.
I see value in especially in the SLAB 2 experiments to tweak the learning later on. So, but maybe you want to start a thread on the forum and when we can discuss. I see value in introducing pieces of learning that we can measure very well. If that can tell us about how, what's the learning computation occurring in the single cell, I think that something we could poke in later on, probably. But at this point, we are focusing on re-implementing existing protocols in the literature.
Yeah,
that was our focus.
Yeah, I understand. It's just that it's interesting to investigate what is the capacity of single cells.
That's
when
right.
They predict, right? After a certain time scale, do single cells are no longer able to represent the prediction, right? And therefore you need some kind of network effect, right? So
it's going to be a bit challenging because the cells we're recording is embedded in the network. And as Lucas mentioned, the input could be influenced by other cells. So we're going to have to be careful on how we do this. Sarah has, Sarah said, I would argue that the timescales are being addressed using the sequential level and temporal jitter. I think, yeah, she's just mentioning my point is that we have over sequential design that have longer timescales. So I think you're thinking along those lines, but yeah. Okay. So it's 9:58. What about I had the few analysis that we discussed, and then some of you just had your name where you see relevant when you want to contribute. It's fine if you want to do all, but I think what will be great is if we get started and then to get your hands dirty with the NWB files. And if you have any concern, reach out. Maybe we can discuss where we are in 2 weeks. Does that make sense?
So, yeah, two quick things. One is maybe we can have separate Slack channels. So Slack turns out to be much better than email because there's no spam filters and anything. So maybe people could group up in Slack channels.
If you want.
The Slack you had. And the other one, I wanted to see how many people are comfortable using Jupyter Notebooks or Colab. It's free, but the benefit is that if we push code frequently in terms of notebooks and make them editable and copyable, it seems that already that has borne fruit, that it just speeds things up rather than just pushing code on GitHub and everybody has their own Python version and all of these issues. So maybe if we stick to notebooks and then frequently release them so that you can have it like Lego pieces, and I can take this from your notebook and this from that notebook, and everybody can just run it, let's say in Colab, that might speed things up.
I mean, I like Colab, and you want to share Google Colab and then have people on Slack and then have people sort of try it out? Is that what you meant?
Yeah. So again, I wanted to, because people have different likes. Some people, they really just like to write their own Python code and run it locally and push it on GitHub. And that's fine as well. It's just more difficult to re-implement that on your own machine usually because of dependency issues and so on. And so Colab just takes care of it. And it's easy to also add a lot of commentary text to the code, right? So that's the other nice thing about notebooks.
Yeah.
So I'm trying to advocate basically that if we do notebooks, I know once the code gets too long, you just have to make something that you push on GitHub, but then you can re-import that to Colab, right? So I'm just advocating if we can maybe stick to that format.
Okay. So I'll help set up a Slack channel. It sounds like people want to use Slack. And then coordinate this way. So and is it fine if we discuss in 2 weeks? Do you feel like some of you will be able to present some first-hand attack at this, or like if I reach out in a week and ask who will be willing to just highlight what they did, you'll feel comfortable with it? I'm hearing Alex.
Yeah, I will look into the oddball. So anybody interested, you can contact me so that we don't cross skis.
Can we make sure the content is expired on Slack? Oh, Spider? What is that? Comments on Spider. Oh, because she's using Spider locally. I see. I mean, for myself, I use Visual Code a lot because I can use the AI agent very efficiently, and so that boosts my speed. You probably can do this in Google Colab. Okay.
Yeah, I can also help. I can also help what I did with your code, Jerome, that if you send me Python code, I can make it ready for Colab, and just then we have a notebook of your code, and so then it might be easier to share.
Yeah, Carter share a notebook, so you maybe want to start with that. He has the newer notebook with the receptor field analysis.
Yeah, so my idea was to use that receptor field notebook and then just get everything rid of receptive fields, but take all the 80% of coding is the pre-processing, right? So it's like, how do I get the data from NWB? How do I make sense of it? How to organize it? How do I get the trials? How do I trigger this? And so that's what I mean. So if you don't have to start it from scratch every time, it can be very helpful.
Okay. I'll try to help coordinate an email. And then next week we'll discuss stimulus design. I think Sarah and Farzaneh and others have started to work on this, so we'll focus on that. Okay. Any other questions before we part? Well, thank you.